Okay, in this video I want to do some more protein structure problems. So here's the question. It says the chain shown below is part of a parallel beta sheet. Draw in the backbone of the three residues of the next strand of the sheet below, the one given. Indicating the hydrogen bonds that are formed, as well as the N-terminal and C-terminal ends of the strand you drew. Okay, so first of all, we should see what kind of information we have here. And the most important information we have here is what's given to us, that it's a parallel beta sheet. And what that means is both strands are running in the same direction. So we have from, let's see, this is the N terminus in this case, and this is the C. And the one we draw is going to also be from N to C. So they're going to be like that. Both are going to be going in the same direction. So basically, if we have the one strand here, we just got to draw the complementary one here. So let me show you what's going on. So I'm just going to start drawing this out. Basically, exactly the same way you see it above here is exactly how I'm going to draw it. It's pretty much an identical copy. And then it's just a matter of understanding where the hydrogen bonds go. So we just start drawing this in, putting the amino acid side chains are not specified in this, they're just called R. It doesn't really matter in this case what they actually are. We're just trying to show the hydrogen bonding structure here and show how these two different strands are connected. So there's that. This is an NH. This is an R. This is an H. And this is a double bond O. And a squiggly line on the end here. Okay, so here's my complementary strand here that's going to pair up with this other one. Now all I have to do is show the hydrogen bonding. So I'll show my hydrogen bond here. Remember, delta positive, delta negative. So these differences are going to form that hydrogen bond. We'll form one over here, and we'll form one over there. So there's three hydrogen bonds, and because these are parallel beta sheets, notice what's going on. The bonds are kind of offset. Remember, I said before that in a previous video about the beta sheet that when you have a parallel beta sheet, that you wind up with this sort of offset hydrogen bonding, which is not as strong as the anti-parallel beta sheet. So that's about it. If I was going, and just, I almost forgot to label the N term and C term, and that should cover it. So we can now move on and look to the next problem here. And this is another protein structure problem. So it says the two sequences below are known to be amphipathic. Which one is part of a beta sheet and which one is part of an alpha helix? Explain your reasoning. For your convenience, 7-pointed stars are sketched below in case you need them. Okay, so we're given two sequences, sequence A and sequence B. And we're told that they're amphipathic, and that one is part of a beta sheet and one is part of an alpha helix. So now it's our job to decide which one is which. So first thing I like to do here is I like to just remind myself of what the hydrophobic amino acids are. So hydrophobic, and those are alanine, valine, leucine, isoleucine, proline, methionine, phenylalanine, and tryptophan. What am I doing there? TRP. Okay, tryptophan. So we have the hydrophobics. Now the next thing I like to do is to look at each strand, and I like to put a box around the hydrophobics and put a circle around the hydrophilics. And that's exactly what I'm going to do with these two strands. So we'll start with over here, we have leucine, so hydrophobic, GLU is glutamic acid, circle, ASP, aspartic acid, that's a circle, lysine, also a circle, valine's hydrophobic, it's a box, glutamic acid again, and leucine, serine, and serine are going to be circles. Lysine is going to be a circle. Asparagine is going to also be a circle. Tyrosine is also going to be a circle. Histidine a circle. Leucine, a box. Glutamic acid a circle. Asparagine a circle. Glutamic acid and valine. Okay, so that's the first one. So that's the first one that we have to cover here. Now, we need to cover the second one, which I'm going to do in the exact same way. Basically, what I'm going to do is just put circles and squares around each of these. So let's do that. So glutamic acid, isoleucine, lysine, asparagine. So circle, square, circle, circle. Glycine. Glycine can also be considered hydrophobic. So, and isoleucine we know is hydrophobic. Aspartic acid, leucine, threonine, leucine, lysine, alanine we know is hydrophobic, so that gets a square. Histidine gets a circle. Methionine we know is hydrophobic. Tyrosine gets a circle. Phenylalanine we know is hydrophobic. Lysine, hydrophobic for isoleucine, and another circle around certain. So if we look at this, we can almost tell just by doing what I did here, which one's going to be part of the alpha helix and which one's going to be part of the beta sheet. Remember I said with the alpha helix it repeats every 4 residues or so. And if you just look at this, we have a hydrophobic here, and we have hydrophilic, 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 hydrophobic. So if we count this out, 1, 2, 3, 4, the 4th residue is hydrophobic, and the same sort of thing is going to happen over here. So 1, 2, 3, 4. I mean, it's 1, 2, 3, 4. You know, it's not quite perfect, but it's within reason compared to B, where we see hydrophilic, hydrophobic, hydrophilic, hydrophilic, hydrophobic, hydrophobic. And then we start to see more of a regular pattern. So hydrophilic, hydrophobic, hydrophilic, hydrophobic, hydrophilic, hydrophobic. And it's that way the rest of the way down, alternating hydrophilic and hydrophobic. And that's kind of characteristic of a beta sheet. Now, I'm not saying this is a foolproof way of doing it, that you're going to get the right answer. You're probably going to want to also plot them on these 5-pointed stars, just to be sure, if this were a test question. But what I'm saying here is this is a really good way to get an idea of what's going on, to get an idea of, you know, which one's going to look like it fits sort of the pattern of a beta sheet and which one sort of fits the pattern of an alpha helix, and that's why I do this. Now, as far as plotting these around the strand, these 5-pointed stars, it's really quite simple. They're labeled A, B, C, D, and you essentially just go down the amino acid, down this polypeptide here, or this peptide, and just write them in. So I'll start with A over here, and I'll write leucine, so L-E-U. And I'll go to B, G-L-U, A-S-P, and L-Y-S, and then valine, and then valine at E, and G-L-U over here, and GLU over here, and then that brings us back to A at this point, which is again leucine, and then B, we got a serine over here, and a serine over here at C, SER, and a lysine over here, and asparagine, tyrosine over here, TYR, and up here histidine, so histidine, and back to A again, so we have another leucine, LEU, GLU, ASN, and rather GLU, and ABs, and over here at E we have again another valine. So as you can see, it's not perfect, and I'll just show you what I have here. So at A, I have leucine, 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 so 3 leucines, all hydrophobic. Over here at B, I have glutamic acid, serine, glutamic acid, so all hydrophilic over here. At C, I have aspartic acid, serine, and asparagine. At D, I have lysine, lysine, and glutamic acid, so again, all hydrophilic. Over at E, I have valine, ASN, and valine, and that's mostly hydrophobic right there. And at F, I have glutamine, I have glutamic acid and tyrosine, rather. And over at G, I have glutamic acid and histidine. So we can kind of see already that there's this sort of hydrophobic region over here and a hydrophilic region back here, part to it. So that's usually characteristic of the alpha helix. So I would be able to say that strand A, at this point I'd be able to say that strand A is part of the alpha helix. So it's part of the alpha helix because we see a repeating pattern. Because we see a repeating pattern every 3 or 4 residues. So every 3 or 4 residues. And we also notice as we plotted this that there's a hydrophobic and a hydrophilic region, which means these are amphipathic. And that's characteristic of the alpha helix. So and another thing to point out, which I didn't look at yet, but I will point it out right now, is that strand B has glycine in it, and generally if you see glycine or proline, those things tend to disrupt the alpha helix. So glycine and proline, I mean, one glycine might not make enough of a difference to say that this is definitely alpha helix, but still it's a sign that it probably isn't, based on the way
this question is designed. So, you know, I mean, I'm not going to plot this other one over here. I don't really think it makes sense to in this video. It's kind of like that's kind of intuitive and simple. You just go around the circle A, B, C, D, and plot it. But we can kind of see, as I said before, from this alternating pattern, hydrophobic, hydrophilic, hydrophobic, hydrophilic, hydrophobic, hydrophilic, that this is very characteristic of what we know for beta sheets. So that's all I wanted to cover in this video. Thanks.